taking genetic therapy treatments would modify my DNA. True, false, and there's also an I don't know. Another interesting mix, we've got 20% have answered true, 60% have answered false, and 20% have answered I don't know. So Tim, would you be happy to talk to this question?
Absolutely. Well, thank you, everybody. That's really interesting. And I think when I pondered this question beforehand, I agreed with the majority that in the majority of cases, the answer to this question is no, that the genetic therapies currently in development aren't generally going to alter our DNA. But I would say some of the later gene therapy approaches that we're probably aiming towards in the long term will. So if I can go through this in turn, we've already heard about some of the different gene therapy approaches. So there's messenger RNA therapy, which both Jamie and Jane talked about. And that certainly doesn't alter your own DNA. It doesn't go anywhere near the DNA. The RNA just gets into the cell, doesn't even enter the nucleus. And as Jane said, is just expressed for a very short time. And you might need to redose weekly or multiple times in a week to get continued expression of the correct cystic fibrosis protein. Then there's other forms of RNA therapy that affect people with very specific mutations called splicing mutations. And those are very short sequences of RNA. And again, they would just be delivered into the cytoplasm of the cell, not even reach the nucleus, and wouldn't affect the person's DNA at all. And again, would need to be repeatedly administered to have their effect. Then if we move on to some of the gene therapies that actually deliver DNA into the nucleus of the cell, many of those, and certainly the ones that have been tested so far, again, don't alter the person's actual host DNA. That they just offer additional DNA into the nucleus of the cell that is then read and RNA, and then a protein is formed. So some people on the call may have been involved in the multi-dose liposome gene therapy study that the UK CF Gene Therapy Consortium conducted a few years ago. And you'll remember that gene therapy program worked. It showed we could get expression, and we could get an effect in terms of lung function. But that was just delivering the DNA coding for the correct CF gene using a liposome actually into the nucleus of the cell. And that DNA would last in the nucleus of the cell for a while, but then get degraded and wouldn't in any way alter the existing DNA that's there. It would just be an additive. So the actual incorrect coding of the CF DNA that is within the person with cystic fibrosis's genome was still there. And some of the viral approaches to gene therapy. So at the moment in the United States, they're testing adeno-associated virus, and there's some really promising results coming out, some of which were presented at the European Cystic Fibrosis Conference last week, showing again that we're getting correct protein expression within the lining of the lungs when this viral cystic fibrosis gene therapy vector is delivered. But that DNA, again, doesn't get integrated into the person's own DNA, and it doesn't alter the person's own DNA. So their original cystic fibrosis gene is still there, but there's an additional piece of DNA that the adeno-associated virus has delivered that produces the correct code for the cystic fibrosis protein. As we move on further through gene therapy, and some people on the call may have heard that the UK CF Gene Therapy Consortium is now partnered with Boehringer Ingelheim to develop and conduct a clinical trial using lentivirus, which will deliver the correct code for the cystic fibrosis protein into the cell. That is a vector that would actually alter the host DNA, because that correct sequence for the CF gene would be inserted into the person's DNA within the lining of their lungs, the cells lining the lungs. And that, in theory, has the advantage that the expression of that protein would last a lot longer than with other viral vectors and liposome vectors. So that could offer great benefit. And then, of course, there's the gene editing therapies, which are probably the furthest away from actually being realized, although have already been used in some other conditions, such as eye conditions and blood disorders like sickle cell disease, with some success. So gene editing therapy is now working in other forms of genetic condition. And that, again, would alter the host DNA because it would cut out the mutated code within the cystic fibrosis coding region of a person's DNA and replace it with the correct code, coding for the correct cystic fibrosis protein. So in summary, I would agree with the 60% of participants that voted for no. But I think in the longer term, we're probably looking for therapies that might well alter the DNA of the person.